Sensory transduction in the retina. The retina contains photoreceptor cells, and there are two types of photoreceptor cells, rods and cones. The rods and cones are located at the back of the retina, behind the bipolar cells and the ganglion cells. And it's actually the back of the rods and cones, the pigment disc, which is at the back posterior most part of the retina, that detect the light. So when light is shined into the eye, it has to pass first through the cornea, then through the aqueous humor, lens, vitreous humor to the retina. It will pass past the ganglion cells and the bipolar cells to the back of the rods and cones, where the pigment discs detect the photons. There are two types of photoreceptors, rods and cones, and rods are designed for night or scopic vision. And when we look at images in dim light, it is monochromatic. They are just shades of gray. But cones are our bright color, also known as photo-optic vision, and that's what we use during the day. Now, the pigment discs of rods are made up from retinol and opsin, and it forms what we call rhodopsin. They combine to form rhodopsin. The pigment disc in cones is also made of retinol, but there are three different types of opsins. So that gives us three different types of cones. Now, one particular cone will only contain one type of the three opsins. The rods have an absorption peak at 500 nanometers, but because there are three different types of cones, depending on each type of opsin, there's going to be three different absorption peaks. So, cones that have an S opsin have an absorption peak at 420 nanometers, M opsins have an absorption peak at 531, and L opsins have an absorption peak at 558 nanometers. Neural convergence occurs when a photoreceptor cell stimulates a bipolar cell. But for rods, which display a high amount of neural convergence, rods, there can be something like 200 rods, and all of them signal only one bipolar cell. So there could be 200 different rods. If any of them gets struck by light, they would stimulate one bipolar cell. And as a result, your brain, when it gets the signal, it doesn't know whether it came from one of those rods or from all 200 of them. And so when we look at things in the dark or in dim light, not only is it monochromatic and shades of gray, it's often very fuzzy because our brain knows of the neural convergence, and so it automatically doesn't put in very sharp edges. Cones can also display neural convergence, but the cones in the macula lutea of the fovea centralis, which remember is where it is ideal to focus images, the cones in the macula lutea of the fovea centralis do not display neural convergence. So we would have one cone telling one bipolar cell, telling one ganglion cell, which would give us a high resolution, sharp image. So the cones that are outside of the macula lutea do display more neural convergence, and so they would give a less clear, sharp image. And we naturally focus our eyes so that the image will be on the fovea centralis of the macula lutea to give us the clearest, sharpest image. Sensory transduction is when we convert one type of energy into another. In the photoreceptor cells, in the dark, the photoreceptor, whether it's rods or cones, in the dark, if there's no light at all to stimulate the pigment disc, then the photoreceptor releases synaptic vesicles of neurotransmitter into the bipolar cell. Now remember, photoreceptor cells are not neurons. They are similar to the ependymal cells that make the lining of our brain. And remember, they help produce cerebrospinal fluid. They are similar to those ependymal cells, but they are not neurons. The bipolar cell is a neuron. It's considered the first order neuron of the visual pathway, and the ganglion cell is considered a second order neuron of the visual pathway. Now the ganglion cell can produce action potentials, but the bipolar cell cannot. So let's look, for example, if we look at rods, if it was dark, this rod would be secreting neurotransmitter, glutamate, from here. The bipolar cell, while it's receiving the glutamate, does not release any neurotransmitter into the ganglion cell. So it is not signaling the ganglion cell. But should light strike the rod, then the rod, once light strikes the pigment disc, the rod stops releasing synaptic vesicles. The bipolar cell stops receiving the neurotransmitter, the glutamate. When there's no glutamate, the bipolar cell automatically releases neurotransmitter to the ganglion cell. And so the ganglion cell, if it gets enough neurotransmitter to reach threshold, will send an action potential to our brains. So in the dark, the rod releases neurotransmitter, and that neurotransmitter stops the bipolar cell from sending a message to the ganglion cell. But as soon as light strikes, then the neurotransmitter stops, causing the bipolar cell to release neurotransmitter, which will stimulate the ganglion cell to send action potential to the brain. And codes behave similarly. Light and dark adaptation we have all experienced, and this is due to bleaching. When light strikes our pigment discs, it causes them to bleach. It causes the retinol and the opsin to have to be reset before they can detect light again. And so it takes a few seconds for that to happen. If there's a lot of light, then more of the pigment disc will be bleached, and it will take longer for it to be reset. So in light adaptation, when you walk out into sunlight, so you're in a darker area and you walk out into a bright area, that will cause your eyes to hurt. Your eyes will squint and your pupils will constrict. During this time, for about 5 minutes, your color vision and the clarity of your vision, or your acuity, will be decreased. So if someone were to walk from a store out into the bright sunlight, that would be a good time to attack someone, because they would be less able to identify their attacker, but I don't recommend that, so please don't attack people. Dark adaptation occurs when we go from bright light to dim light. So when you're out in the bright sunlight and you walk into something dimmer, even though there's lights on, sometimes it's almost as if it's completely dark. Or let's say at night, it is bright light because you have your light on, but you turn off your light or your lamp, and now it's completely dark. That is dark adaptation. So what will happen is your pupils will dilate to allow in more light. The rod pigments have been bleached by all of the bright light, so they will take a few minutes, up to 20 minutes, for them to become unbleached, for their rod opsin to regenerate. And so after a few minutes, you'll start to be able to see fuzzy shapes, and after about 20 minutes, then you can see as well as you're going to see in that dim light. The dual vision system is our explanation of why we have rods and cones. Rods allow us to see, not very clearly, but they do allow us to see in dim light. So they're our night vision. Cones allow us to see clearly and accurately during the day, but if we had cones alone, we would be blind at night. So the duplicity theory says that one signal type of receptor would not work well in both dark or dim light and bright light, so we have two sets of photoreceptors so that we can see in two very different types of lighting situations. Now, in order for us to see color, that's where these three different options come into play. Because we have three different absorption peaks, that allows us to detect three different colors. And that seems odd. I always think, I understand intellectually how we see, but whenever I'm blending paint colors, it would be different to associate blending paint colors, in which if you took some red and some yellow and made orange, that is a different method. That is not how our eyes see color. What actually happens is different photons at different nanometers, different wavelengths, have different energy levels which we detect. And so at any given time, many photons are striking our cones, and that's causing our cones to tell our bipolar cells, which tell our ganglion cells, which tell our brains. And so we're looking at the ratio of the cones that are stimulating our eye. For example, if only S cones are stimulated, then we're going to interpret that as violet. So if we see a block of color or a block of light, let's say someone's sweatshirt, and all of them are, and only in that area where that light is coming from, only S are shining through, we're going to interpret the color of that sweatshirt as violet. Now if there is some S's, lots of S's stimulated in a few M's, then we're going to interpret that sweatshirt as being blue. And if there is S's and a whole lot of M's and L's stimulated, we're going to interpret it as blue-green. And so this is why dot matrix printers work. You know that whenever you fill the ink for your color printer, you only put in 3 different colors. And what it does is they just form different little dots of color in those 3 colors. But because those dots are in a certain ratio, as your eye detects those colors in that ratio, we interpret it to be a certain color. So it isn't actually using those 3 colors to mix it into, let's say, orange or purple or green. It's actually just forming dots that have the correct ratio, which cause our brain to interpret it as a particular color. So that is how color perception is based. Now, color blindness is hereditary, and it is more common in males because it is an X-linked on the X chromosome. And so since males have one X and one Y, they're more susceptible. But should a female have 2 X's and both X's lack the ability to form all 3 cones, then they would be colorblind as well. But it is way more common in males. Stereoscopic vision is depth perception, and in order for you to have depth perception, you have to have 2 eyes so that you have overlapping visual fields. and your brain interprets the angle from one eye and the angle from the other eye and realizes where those angles meet, and so it interprets that something is closer compared to something else. But we need to have two eyes in order to have stereoscopic vision. So this leaves us with the visual projection pathway. Now remember, the photoreceptor cells are not neurons. So our first order neuron is our bipolar cell. The bipolar cell will stimulate the ganglion cell when the rod or cone stops stimulating the bipolar cell. Remember that? So the ganglion cells, the axons of the ganglion cells, make up the optic nerve. And so the axons of the ganglion cells on this left eye are red, and the axons of the ganglion cells in the right eye are green. And so the optic nerve, there's an optic nerve, cranial nerve 2 from the right eye, and cranial nerve 2 also from the left. They form what we call a hemidesiccation at the optic chiasm. The optic chiasm is where half of them cross, but half of them stay. And so if you look at my picture here, you will see that 2 of the green fibers cross to the left, but 2 of them, they go into the optic chiasm, and then they go out, and they stay on the same side as the eye that they originated from. And the same thing happens to the red fibers on the left side. Once the fibers leave the optic chiasm, where they hemidesicate, the fibers are known collectively as the optic tract. They will head to the thalamus, because second-order neurons head towards the thalamus, and they will synapse, and the third-order neurons will leave the thalamus, and they will travel to the visual cortex, the primary visual cortex, in the occipital lobe. So that is our visual projection pathway. The ganglion cells make up the optic nerve, which travels to the optic chiasm, and half of the fibers cross and the other half stay on the same side as the brain. And they leave the optic chiasm through what we call the optic tract, travel to the thalamus, where they synapse with third-order neurons, which continue to the primary visual cortex in the occipital lobe.